So it's my pleasure to introduce Reid, co-speaker of the morning play. He's at Berlin Institute, was previously at Harvard, so he talks about computational geometry of cortical networks, manifold-embeddings, analysis of multiple-site recordings, connectomic graphs, and deep learning networks. Please take it away.
Thank you. Does anyone have any questions? This was, you always get badgered for titles, and sometimes you say enough. Okay. Here. How's that? These are ideas I've been messing around with for well over a decade, and they were kind of mature and extraordinarily difficult for me at the time to explain. But they're ideas that require a very nice connectome to put into practice. So around 2005, we started doing EM connectomics. I started playing around with these ideas, and maybe it's finally time to finish these ideas because we're really getting to the point where we might have big enough graphs to test them out after more than a decade. So these are two things that one has when you study the brain. One thing you might want to get when studying the brain, what are representations in the brain, and very obviously connectivity with help from biophysics and dynamics gives you physiology, and also just as obviously physiology overdevelopment and even in mature brains through plasticity gives you connectivity. But what about representations? What about things we might want to understand in finding out the purpose of a network and of individual cells? Classically, one looks at inputs in sensory systems, behaviors in motor systems, or combinations of the two. And through physiology, by looking at input-output relations, you get things that you might want to call representations. For vision, it can be X and Y or theta. And really not just those are the parameters, but these are what receptive fields typically are functions of X, Y, theta, velocity as you percolate through the visual system. The parameterization might be gaze angle for face areas or face identity. Are there other ways of getting at representations? And it's satisfying to me that so much of this workshop is about how one can go from physiology to representations through dimensionality reduction, manifold embedding, etc., through various forms, via various forms of correlated activity. But how can one go from connectivity to a representation? And maybe this is something that is obvious, maybe it isn't, but I haven't seen so much in the literature about this, and it's something I'd like to explore in the next hour. What can we learn about a representation in the absence of input-output physiology? Again, from physiology correlated activity. I'm not going to talk about dynamics and trajectories the way so much, Ila's talk and other talks you'll hear, but just generalized correlated activity. What can one do with connectivity? Can dimensionality reduction be used to understand connectomes as well as multineuron physiology? Can the matrix, connectivity matrix, Wij, be used to infer the geometry of a representation, and not just the geometry, transformations on representations? and identically, pretty much, the way one can look at correlated activity to get a representation, one can look at correlated connectivity. So, and this is pretty much the story of my life. Back more than 20 years ago, the question I was consumed with is, how does visual physiology relate to single thalamocortical connections? And based on the Hubel and Wiesel model of connections from visual thalamus to visual cortex, there is a very simple way that the labeled lines in the thalamus might connect to labeled lines that the neurons in visual cortex with a certain physiology. And in those days, we studied connectivity with temporal analysis of two spike trains, doing cross correlations and looking for a delayed peak in firing after the putative or potential input cell fired. If there is a delayed peak in firing like that, that is very strong evidence of connections. Let me introduce some variables, because they'll be throughout the talk. The Hubel and Wiesel model is about connectivity on the one hand, and very, very separately, representations. And let's label the units. This is a simple cell, which a simple cell is a bar detector that, for instance, we'll see a bright bar on a dark background in this case. So that's simple cell 1. That encodes a representation, and that representation is it responds to light bars on a black background with this certain oblique orientation. It gets inputs from LGN cells. So for non-visual physiologists, L, see it as an I almost, it's the input layer. S is the oriented filter, very much like a wavelet or a Gabor function, or the first layer of a convolutional network. So these four inputs, L1 through L4, that have these four representations, they see points of light on a dark background, and those points of light line up along where the representation of the target is, and that's the model. Couldn't be simpler. Over the ensuing 20 years, we essentially, or 15 years, I guess, starting 15 years ago, my group just geared up techniques to get at the generalized question, how can you study the function of cortical networks, which are massively interconnected, and these correlation techniques do not work. How can you study the function of a network and the connectivity of a network? And first, we applied the fairly new tool then of calcium imaging in the cortex to watch, rather than listening to neurons fire away, watching them fire away. I won't show it, but you can watch 100, now 1,000 neurons at a time responding to stimuli, and you can map what they do. 5 years after that, we started to work on connectomics, on large-scale electron microscopic EM reconstruction of circuits. And this is the second of two papers that we did. Kind of hard to see, but maybe that's the point, that here's a huge network of cells. This is 100 neurons that reconstructed the axons of about 30 neurons, the dendrites of about 100 neurons, and mapped the connections between them. But we labeled those neurons according to what they responded to, the orientation that they best responded to. And did a ton of work, and found the general simple idea that a priori one would say that has to be true, but 10, 15 years ago, dogma was that the cortical circuit was random with respect to function. It was some magical pruning of weights or just dynamics that gave you it. Silly, but that it was nice to have a straw man for all those years. But really, it's proving to be true that the cortical network is similar to the thalamocortical network, like-to-like connectivity of cortical neurons. Red neurons respond to horizontal, green to vertical stimuli, and those form these privileged networks, subnetworks of like-to-like connectivity. So cortical functional connectomics, we call it, we've been doing for about a decade now, and there are really only two papers in this field. One we did in 2012, and one in 2016. You know, this is not a fast, quickly moving field. The first study had 200 connections, roughly 1,000, in less than 100 neurons. The second study, which really gave the like-to-like, was 1,000 connections amongst 100 neurons. What are we doing now? We have this huge grant from IARPA, so our group, myself and Nuno da Costa, doing the electron microscopy, Sebastian Sung doing the segmentation, and you'll hear a lot about segmentation from Varen later on, Andreas Telius doing the physiology. What are we doing? We're reconstructing a cubic millimeter, about 50 times larger than what we had done before, and with the magic of postmodern segmentation of these data, we expect we will study 10 to the 5 neurons in this cubic millimeter, and we expect somewhere between 10 million and hundreds of millions of connections in this connection matrix. So what can we do with this huge object? Just 2 minutes. I'm going to allow myself 2 minutes on experiments. I was going to show nothing, but someone said, you've got to show something. So how are we going to study 10 to the 5 neurons, 1 billion synapses in a volume, 25,000 40 nanometer sections? In order to make a 3D volume, we need to take 250 million images, 20 megapixel images, put together into a 3 or 4 petaboxel data set. And we're doing that with a bunch of early 3, but now 6 electron microscopes that are sort of tricked out in a way that we started first at Harvard more than a decade ago, to make this infinitely big data set. Let me very quickly, Varen will, oh golly, I'm going to have to go through my browser. Ignore everything. Good. There we go. What does the neuropill look like? This is one electron microscopic figure, and I can zoom in on the left, and this is what electron microscopy doesn't look like. All the grayscale stuff is what electron microscopy looks like. And you can see these long things that are dendrites, another dendrite, another dendrite. You zoom in some more. You see a bunch of circles there. Those are vesicles. There's a synapse near there. I'm not going to find it. But this is colorized by these convolutional networks that Sebastian Sung and his colleagues, Varen Jain, who's now at Google, and you'll hear from him, have used to trace these neurons by machine. And it's nice to be able to colorize one section, but it's extraordinary to be able to, these are serial sections. It's kind of fun. This is what the fun of serial section EM. You can zoom through in 3D following these things. And the convolutional networks follow in 3D these shapes. And one nice feature that I was thinking about a lot yesterday in Terry's talk are apical dendrites of neurons. And another feature of apical dendrites, these sort of antennae that come out of neurons, is that they go in bundles that some people didn't believe in, but here I will prove to you that apical dendrites go in bundles just by clicking one. Let's click another. Let's click another. Let's click another. That was about 6 months of a postdoc's life previously in doing, and Terry knows this, he's done. I was just talking to Tom Bartol, who wants this. Tracing these things in 3 dimensions. There's one that has a cell body. Here's another one, maybe here.
What's the spatial scale here?
That's 140 microns. But imagine this embedded in a cubic millimeter. It's infinite. So this is a fun tool that was built by Viren's team at Google. But the star of this show here is the segmentation that Sebastian Sung's team has done, and that Viren is doing very similar at Google.
So Clay, are these really cluster bundles, or would there?
It's clusters.
It's clusters.
It's
there's
clusters.
Then some space where you don't have these bundles.
Yeah. I mean, they're everywhere, but they're especially dense in these clusters. So let me go back to the real talk. What might we learn from a cubic millimeter? It's a really obviously dense network, but it's also a hierarchical network. Don't ask me, we can talk later, how you can get all the thalamic inputs, but you essentially trace them from where they enter the cortex, and then you have a label on it. To layer 4 of the cortex, recurrent connections in layer 4, to layer 2, 3 of the network with recurrent connections, and then on down to 5 and 6, and there's a feedback this way. It's a huge hierarchical network with lots of recurrent connections. Let me just concentrate today from thalamus to layer 4 to layer 2, 3. And for the sake of argument, this is all physiology and anatomy, or physiology from the cat, but not the mouse, which we're doing now, but let's leave that alone. So this is equivalent to understanding the transformation from point detectors, center surround detectors, to these localized oriented filters, these wavelets called simple cells, through the next layer, which is complex cells, which I'll talk about, essentially bunching together neurons, grouping together neurons of the same orientation. But it's also extremely similar to any hierarchical or any convolutional network for vision. There's the input layer, the images. There's the first convolutional layer, which is always these wavelet-like things, like simple cells. And then the second layer, which people really, they throw up their hands and do all sorts of visualizations for. But to me, and actually looking at it in detail, it has a lot of the flavor of a complex cell layer, and then all the way through.
So actually, to go back, there's between the slab labeled S and C, there's actually another set of cells called, max pooling.
Yeah, there's max pooling. And
those really, I mean, it's equivalent, I think, to a complex cell. The max pooling.
Yeah, potentially. So yeah, but that's so trivial, I haven't analyzed it. You know, it's part of the model and part of, so yeah, maybe it's a complex cell.
Well, the original method,
no, it isn't. We'll talk about it. No, it isn't. It's so simple because it's taking one of the 96 kernels here and doing a 3x3 max pooling. And that's not what complex cells are. Complex cells will take all comers, different simple cells of different.
Right.
So, and that's this layer. So there's sort of there's an output nonlinearity in translation, you know. Right. But so the analogy breaks down. But I do think conv2 is closer. So can we do this? Yes, probably. And Davi Bakanai, who started doing the Connectomics in 2006. His first thesis project was doing exactly this in the pathway from retina to thalamus. But these ideas, given dense enough reconstructions, this should be possible for a visual connectome. And the thalamic input could allow a foothold to interpret cortical networks. So let's do a computational experiment. And despite the title, I'm going to spend most of the time on a hand-built cortical network. Spend the last 15 minutes, 10 minutes on AlexNet, a convolutional network, and then just the last slide, which is sort of, well, the last slide is doing the physiology experiment to match the anatomy experiment. And I was going to skip this slide, but let me do it very quickly. A local patch of visual cortex, it's really important to I know you have issues with this, hence the next slide. It's very important to be careful to not mix up brain space, where things live in the brain, with connection space, a connectome, or a graph that can have a layout or not, and function and representation. In this case, this picture is actually a functional picture. The different colors express the function. So, and this is really the theme of the talk. Physiology will help us understand wiring diagrams, and wiring diagrams will help us understand the physiology. Here, emphatically, I'm talking about local networks, and there's obviously a continuum between local connections and maps and something called functional architecture, which is sort of a micro map. What I'm talking about is sort of like in the ConvNet, all of the weights that are trained, but not the weight sharing that happens all over the network. And in Cortex, there are maps of functional architecture and that take on several dimensions. They take on X, Y, theta, maybe stereo. But local networks have hidden dimensions. And those hidden dimensions are the receptive field attributes, phase, and all sorts of things that have to do with different receptive fields, different function, all mixed together in a salt and pepper manner. Orientation is mixed together in the mouse visual cortex in a random fashion. And it's kind of a dumb analogy, but I still like using it. It's very similar to the physical world, where we live in a easy to understand macroscopic four-dimensional manifold. But go down really small, and there are a bunch of hidden dimensions, 6 or 7 hidden dimensions that in a local cortical network, there is space, and in fact, it happens that there are hidden dimensions of representation, all the way down to the single neurons that encode dimensions that aren't visible macroscopically. So here's the model. Let's take an array of point detectors, these input or LGN, that are just Gaussians in space. Let's make 256 cortical cells, which in the simplest case are just elongated Gaussians. They can have side lobes and stuff, but it's easier to discuss this case and easier to compute, but you can do both. So you have the representation, which is a bunch of points in space, point detectors, a bunch of point and orientation detectors, 256 of these orientation detectors at different locations and different orientations, 14 by 14 grid of these points. The size of these filters are wrong. You can see they're this size, so you can see them all, but the actual filters are big on this grid. And the rule is very simple. The Hubel and Wiesel rule that we worked on, it's essentially the strength of connection is proportional to the overlap of the two receptive fields, the dot product. So the weight matrix, Wij, is proportional to the overlap of the two representations. The input receptive field, the LGN receptive field indexed by J, and the simple receptive field, the oriented wavelet receptive field. But essentially, the weight matrix is equal to the dot product of the representation, of the two representations of the two things that may or may not be connected with a certain weight. So given this simple linear relationship, what determines the connectivity? That determines the connectivity. Can we infer the representation? And this is either trivially obvious or it takes a bit of explaining, which I'll do. But essentially the idea, this is so overdetermined. You have a two-dimensional manifold and you have hundreds of functions on those two-dimensional manifolds that have very simple rules. Of course, you can reconstruct that manifold. In the very simple case where everything's on the grid, you get this weight matrix of the input dimensions, 144 by 256, and it has a diagonal structure. It should be that way, but that's the way plots are shown. Anyway, step one, find the latent variables that are similar to the receptive field centers of the inputs. So, so we're trying to get the best parameterization of the inputs. And the insight is sort of obvious. If LGN receptive fields are close together in space, they connect to the same targets. So that will allow you to make a pseudo distance between the two LGN cells. And just to overdo it, but I always, whenever I give a talk like this, I underdo it and people don't get it. But for this crowd, this might be a lot of overkill. You just take the covariance matrix of the LGN to cortex connectivity. Essentially, you do Wij transform times Wij, and you're left with a covariance matrix that has the dimensionality of the inputs. 1 minus that will give you something that looks 1 plus that, anyway, that looks like a pseudo distance. No, it goes to 0 for far away, for nearby, sorry. Does that make sense? I sort of muffed it in words, but it's very obvious.
How do you get a distance greater than 1?
Good question. I felt proud that I figured this out myself, and I discovered, oh my God, everyone knows this. But anyway, that's what I'm doing right now. Here we have a secret. This is the actual distances, and those distances range up to a certain amount. There's 0 along the diagonal and can get very far away for in the corners. It doesn't look anything like, or it looks a little bit like the correlation 1 minus the correlation matrix. But yeah, everything saturates greater than 1. So I scratched my head for a couple of years in the time scale of this long-term project, and I read, oh, Dijkstra algorithm, Wikipedia or something. So if you follow that with a graph Dijkstra algorithm, sort of integrates out the local distance, and you get something that looks identical to the actual distance matrix. Wow, fantastic. That's precisely what isomap is anyway. So I did isomap on this, which is essentially take the pseudo distance, integrate out that for things that have distance of 1, sort of the asymptote, and you get something that's just like the actual distance matrix. And then you do multidimensional scaling on it, and you get exactly the right answer. This is what, from all this, the answer is this. You start with the grid, that's secret, but scramble everything up, and still you reconstruct the grid just by doing this trick of essentially finding the correlation coefficients of the weight matrix, the same way you'd find the correlation coefficients of a physiology matrix, rather than summing over T in the correlation coefficient of physiology, you sum over the cortical targets. And you get this, and it works beautifully. Here's the slightly hard next part. So we've gotten the manifold of the input space, and that's kind of exciting and could potentially generalize. To the extent that the inputs to any cortical area look like a manifold, sort of non-categorical, smooth, parameterizable. You might be able to discover that in a word area where we know from networks that semantics live in a local vector space. This might be kind of general. It might be a general cortical feature. So you can bootstrap your way into the representation of the inputs, then take that bootstrapped representation and just apply W, the weight matrix, onto that representation, and you can get the representation of the next level. So essentially, these are the inputs, but those are invisible. They're long gone. All you have are the weights. And applying the weight matrix to the manifold embedding of the LGN cells, you get something that looks almost, well, doesn't look almost identical. It looks highly similar. Matching. There are edge effects here, but let's look in the middle. And now, like Ela, I did I allowed myself a mirror reversal and a single rotation, and the matching inferred bootstrapped simple cell to that is that to that. So you can get into the network. And now I always in the past lost people there, so this really goes through again. So we have an input space, the LGN cells, L1 through L4. We have the output space, the S's. We have the connectome Wij. What I've done is bootstrapped into and got LRF of X and Y, and applied W to get the SRF of X, Y, and sort of theta. And let's even keep going. So the mapping from L to S is W, and these are what the elements of L and S. And here's what it looks like. You know, again, this tells you what the players are, and you have this nice diagram that you see from your youth in differential geometry or something. The connectome is this thing. It's just the mapping, the linear mapping. This is the fun part, doing isomap or MDS, multidimensional scaling, and get a sort of pseudo projection. It's not exactly a projection, but of the L space to the same units, L, but they're transformed now to be functions of X and Y. You discovered the X and Y parameterization of these things induced by the wiring diagram W. And now it's sort of simple. If you have the same L's just with an XY label and you apply W to it, you get the S's. And that's what this trick is. You have the parameterization of your space. W selects the inputs and gives outputs. Okay?
Question.
Yeah.
So when I stand correctly, though, this is about the centers of the receptor fields. It's not about the shape.
Precisely. Yeah. So, but that's the fun thing. So, you bootstrap your way into exactly that. I should have made it more clear. Into inputs that are points. Absolutely, are points in a manifold, and really what are receptive fields and things, they're functions on manifolds. But as long as the parameters, well, as long as the inputs are simple enough and just the location in parameter space, it could be X, Y, theta phase for simple cells, for instance, that might be good enough. But once you're bootstrapped in, then you get functions on the manifold. The S is a function on the manifold. But yeah, right. So don't trust me, I wouldn't, because I did everything on a grid. I'm going to just rush through this. You can randomize everything and have random locations, random orientations, etc. You get a connection matrix that looks like nothing, but still you get the right answer. These are where the inputs live, and those little tiny tails are the errors, and you can do the same trick. So what about the second stage? What about, so what I did was the thalamus to layer 4. What about layer 4 to layer 2, 3? And again, there was this 50-year-old model from Hubel and Wiesel that there's things called complex cells. Think of that as conv2 in a convolutional network. And this is essentially lumping together a whole bunch of different wavelets, all the same orientation, different space, different phases, etc. So we have S's mapping onto C's. And very quickly I'm going to go through this model where you can have the inputs that, again, are different positions, different orientations. The outputs are now these much larger spatial things, but they have different orientations, and the rule is grab from nearby orientations. So this is just building, building out the complex cell model, and the color code here is different orientations. Okay, build up that model and then do exactly the same trick, and in this case, the inputs are functions of x, y, and theta, and we have a different diagram. S to C through this V weight matrix, project it, the same S's onto a parameterized set of S's, and then you can get your complex cells. But the important thing is finding the parameterization of the inputs, and it gets the next slide is a little bit complicated. I use color here for the orientation. Forget that. In the next slide, the color code is X and Y. Brightness is the Y dimension. X is the color. Okay.
So far, this is feed forward.
This is feed forward. Yeah.
What about the recurrent connections?
You get all the same principles hold, and you can't do the trick. You can't do this trick because both sides are the same units. So you can just get the parameterization, but it works pretty nicely. And you can go backwards, and you get the parameterization of the target space, not just the input space. It generalizes completely, and essentially the whole thing is not I by O. It's I, layer 1, layer 2, the entire matrix. But it's sort of nice to do the embedding cell type by cell type. So anyway, you can embed this model into 4D, and it's hand built, but it's just kind of fun to do this, and you really get R2 by S1. You get a three-dimensional manifold that you have to do a 4D multi-dimensional scaling, but as you rotate this four-dimensional point cloud, you can find the angles where you get this, the orientation direction, and if you get it just right, along another projection, you get retinotopy, Y and X. So it works for decently complicated manifolds. In a way, it's all a parlor trick that it's built that way, but it's fun to see that it actually can work. In the last some number of minutes, I'm going to apply the same ideas to AlexNet, which is one of the very first highly successful uses of a deep network, specifically to work on ImageNet, this huge image data set for classification. But it's a N, 5 or 6 layer convolutional network. They're color images, and you get color first layer convolutions. But I had a friend, Gayatri Mahalingam, do a monochromatic AlexNet. She just did everything in black and white, one channel rather than 3 channels. And so we have a bunch of these wavelet-like things in the first layer. And let's do exactly the same thing. I'm not going to show you that it's really kind of trivial going from input space, essentially pixels, which you can scramble. And you unscramble the pixels when you go through this manifold embedding stage. But let's look at the interesting stage. What is conv2? What does it look like? And most importantly, what's the as seen from conv2, what is the important manifold that conv1 lives on with respect to grabbing things that are similar. That's what it's a function on the manifold, and continuity or something will give you that things that are similar need to map to the same place. So again, the input is like the LGN, the conv1 wavelet is like simple cells, conv2, people really haven't said much about it, but what does it look like? I've said this 100 times, so conv1 to conv2, what is that mapping? And here we go. There's so many weights and so many, so conv2 is quite large. It's the spatial kernel, the spatial support of these filters is 5 by 5 by 96. It's not, there are two of these banks, so 96 deep. So it's 96 times 25, whatever that is, 2,400,000. Elements that go into each one of these. And looking at all of the potential inputs, you get something that looks very much like a ring. It's very small, but can people see? I'm going to go to the next slide. There are many versions with different parameters. You can take the inputs that map onto a manifold with the least stress. So, but there are all sorts of selections that you can take to try to uncover the shape of the manifold. And here's a nice one, which is just abundantly clear, that the inputs cluster into something that looks like a ring, which alone you'd say, yeah, maybe, but here are the actual kernels, the wavelets that are at different positions in this one-dimensional manifold shown in 2D, or all the verticals here, all those obliques there, horizontal, the other oblique. So clearly, conv2 layer cares most, in some sense. The rules of connectivity respect the proximity rule as you would get with S1, just a circle. To do next, and I've been playing with this for a while, to see if it looks like R2 by S1, and it does, but one has to sort of unpack the weight sharing and let's ignore that. Almost done. This is my one slide, and there's not much. I'll walk through it. This is exactly the same thing. So we have a plot of a bunch of the input filters plotted in 2D, and the 2D position of these filters is determined by manifold embedding. In this case, however, the manifold embedding is not acting on the graph, it's acting on the physiology. So again, Gayatri gave me the activations of the first convolutional, the output of the first convolutional network, the activations of the second convolutional networks to 300 different images in ImageNet. And there are a lot of numbers in that. It's a 27 by 27 grid of points in that convolution, and I'm treating those as independent. You're either different neurons, or you can better, for this thought experiment, consider them the same neurons seeing an image where you move the image around. So you have 27 squared positions times 300 images. It's a lot it's a very long temporal vector or activation vector. And you do the dot product of those activation vectors and you make a correlation coefficient matrix, again, 96 by 96 of the inputs.
So it's a lot easier than having to record that much.
It's a lot easier. I mean, that's the beauty of this, and Anyway, so the answer looks the same, and the tendentious point I want to make is that, particularly because of the sparsity, I think, of the code, they're very high biases or low biases, depending on how you say it, but these neurons don't fire very much, and some of them fire almost not at all, these units. It's a noisier convolution, I think.
So do you think that it would get more precise if you had more examples, more inputs?
Probably, and it's an easy thing to do. I just have to ask Gayatri to do it, and she's busy. It would certainly. I know I've got to set it up myself. If I put all of the biases to 0, then it would be a very dense signal, and it would be probably much better. So I'm essentially done. Given like-to-like connectivity, given just the simple postulate of a Hebbian learning rule, if a representation lives on a manifold, we can infer that representation from the connectome. So that's the take-home point. And putting that into convolutional networks, I think it's an interesting, I don't really know, but it seems like to me to be an interesting research question. Where in a manifold, where in a convolutional network do things appear not categorical or chaotic, fractal, or whatever? When do they appear continuous manifold-like, and when do they not? And my guess for cortical networks is we already have maps. We already know the cortex likes continuous representations. My guess is that, the super guess is that a cortical column is a principal component finder, and it picks out things that look like directions, points in a manifold, and operates on them. So this might be very general, not just in early sensory systems. Potentially, we can take a connectome and bootstrap our way into understanding the underlying representation. So it's not just embedding the inputs into a manifold or embedding anything into a manifold, which by definition gives you points in the manifold, not functions in the manifold. You have the transformations in the connectome as well. So that's why it's arguably richer. But Mitya will disagree tomorrow, I bet, or today. Arguably richer than doing it with physiology, because you have the transformations built in.
What about combining the two?
Yeah, yeah, absolutely. I mean, this started from doing functional connectomics, from doing physiology and connectomics together. But I just, said, let's be cute and let's try, to imagine what you'd do if you had a Broca-Wernicke connectome. What would that look like? In AlexNet, we can go from conv1 and from input to conv1, or conv1 to conv2, and show this manifold embedding ideas work. And essentially, the idea is this: a connectome is a mapping, and it can be completely chaotic, and doesn't need to be on a manifold. But in the cases I looked at, you can embed it in a manifold, give each unit a parameterization, and then operate on it by the connectome itself. And you can even do this at multiple stages. So if you have all of this in one piece, you can go from L to S, S to C. You can go from S, X, Y to V. You can add a projection from here. There are a lot of directions you can go in this graph crawling through manifolds. So
why stop there? Go from conv2 to conv3.
I have, and it's a god-awful mess, and I can't figure it out. And this is a hobby. I just like, it's sort of programming therapy. So these are the questions. In which cases do representations live in a manifold? primary sensory motor, certainly, but what about hidden units, funky things in the middle? How often is that representation derived from the connectome? So this is a question I was talking to Ela last night. Can dynamics live on a low D manifold, but not in the connectome? I think generally not. I think the connectivity has to embody the manifold, but who knows? If you can modify weights without modifying connectome proper, who knows? And this is Tara's question. When is physiology alone sufficient? Best for uncovering the representation, its manifold, anatomy alone, or obviously both. So these are ideas I started doing with Davi Bach, experiments, starting with Jose Manuel Alonso and Marty Usry for the Jubal and Wiesel-like experiments, and then Davi Bach, Wei-Chung Li, and now Nuno da Costa on the Connectomics, and discussions with these people. Thanks.
So I want to come back to this question, if you have recurrent circuitry. And so the question is a little bit, so as long as your neurons live as points on these parameter manifolds, that kind of means that there is a feature of a single neuron's response. So you can kind of, you can assign an orientation to the activity of one neuron without looking at other neurons. But if this goes out of the window, so if you have extra classical receptive field properties of neurons, or so my question is, if the neurons stop to live as points on the manifold, but there are some manifolds maybe, but they have to be described by populations rather by single neurons, how would this theory generalize?
I have similar answers to this question and questions about the science that I like to do of feed-forward connections. I believe, along with Murray Sherman and a lot of people, that cortical networks, in particular thalamocortical and cortical networks, have a feed-forward backbone that the things that can make the neuron fire, everything else being, the data or whatever, it being in the right phase, the right things happening in the surround, et cetera, et cetera, et cetera. I think all of these manifold embedding things are finding the backbone geometry. And all the other stuff, I think it's extremely complicated. And arguably, I think it's stuff that you might be able to get with the connectome. But then, so that's my hand-waving way of saying, I'm putting that off, and I don't want to, I don't understand how to answer that question, but I think the backbone is important.
But then, in a sense, then your summary should include the words feed forward, right? Because after
absolutely.
all, in physics, when water is something where the interactions between molecules are determined, but then it has all these states of matter, right? There's steam, there's ice, there's liquid. And all of this is dynamics, and
absolutely.
The dynamics that emerge, part of the study of neural circuits is indeed, like in the sensory pathway, there's a lot of feed-forward processing. But in other cognitive circuits, we also know, like in memory circuits, a lot of our models basically depend on emergent, stable states from recurrent processing, and that really is the fundamental representation. Is this emergent response, right? So
I'm with you completely, and in my own brain, and people who know me can have a little disclaimer up here. Clay likes feed-forward networks. And the standard question is Fritz's question, but the next question is your question, dynamics. This is static. This is static, and you might be able to get dynamics from this, but I'm not counting on it tomorrow. Yeah. So it's a regularity, but it's necessary, but not remotely sufficient. And sometimes it might not be necessary. Sometimes there could be recurrent dynamics that come out of, from my point of view, nowhere. So absolutely.
So this
So
is
yeah.
an inference step, right? One has to just, I mean, I think the problem is symmetric in some sense. You can go from connections to dynamics, to activity, but it requires this inference step where you have to plug the connectivity into a dynamical model, and then
Yeah.
get the emergent, dynamics by scaling. And similarly, the other effort, of course, until your beautiful work and others, work from Sebastian and others to get connectomes. Until then, we were sort of stuck with trying to infer connectivity from dynamics.
Absolutely.
And that required this inverse inference step. And I guess each one has its own issues, I guess, this inference or inverse.
Absolutely. And, I think different places, different principles. In places where inherent dynamics find the attractors that are memories, sure. But yeah.
So, just to follow up. So, I think you have to distinguish between, in terms of the actual function, between patterns that you're very familiar with, right? That you see faces all the time, for example, and you recognize them really quickly. In fact, who was it that did this experiment where he showed that if you take things like Eiffel Tower and so forth and look at the responses of the output, it's almost there's not enough time to do any processing except the feed-forward pass, and then you get the output. But as soon as you give someone a more complex image to analyze, then it takes longer because it does have to circulate a little bit longer for the computation to settle. And by the way, this is Jim DiCarlo has done followed up on his original work, which we heard about on Monday morning, the first talk, which has to do with looking at comparing the time it takes a monkey to get the right answer and whether or not the ConfNet, AlexNet, gets the right answer. So it turns out if you take the images that AlexNet gets wrong, it takes the monkey 30 milliseconds longer to get the right answer, suggesting that's what's going on functionally. In other words, the network has feedback, it has lateral interactions, and you really need that to do the hard problems.
Yeah, I believe it.
So actually, I have some question about the foundation of this question about connectomics, the importance of connectomics. Because from our understanding of, let's say, artificial neural network, I'll talk about it tomorrow a little bit, there are actually infinitely many random configurations which are as optimal as far as achieving the goal. And it's really a fallacy to talk about one optimal brain, and in the terms of the connections of the synapses. And I wonder if you have any way of actually comparing two different brains in the same tissue, let's say, and see how similar the connections are.
Well, there's a lot there. So let me there's a lot there. Clearly, the individual things are going to be different. I would wager in these that the underlying manifold is going to be the same, and maybe Mitya will talk about similar ideas. Yes.
Maybe.
Maybe. Okay, I'm sorry to keep putting words in your mouth from a 4-second conversation. However, I want to strongly disagree with the idea of optimality and equivalently useful. I never claim this is optimal, but I might claim that evidence is pointing towards this being true, so in the network. So it won't be true everywhere, but empirical results about the connectome, empirical results about physiology and representations, those are what they are. And nature could have solved it a whole bunch of different ways, but my big belief is that the cortex solves it in one way, and particularly these sort of overtrained parts of cortex that have a lifetime to develop. I do think that they have Hebbian rules, i.e. like-to-like connectivity, and they seek out and operate on low-dimensional things that can be transformed by finding these correlations in the input. But you know,
local organization can be very, very different.
Of course, it can be different. But what I'm saying is, when you look at 1,000 cats or mice, you find this, and every cell is different. You know, it's biology, but the underlying rules, i.e., locality in space, tuning for orientation. Those things are remarkably constant.
I don't argue about the rules. I think the principles are important and are general and universal. But the details of the connection is just like trying to find the detailed configuration.
Well, no, but this I am going to keep arguing, because for whatever reason, the strongest complaint that people have about connectomics is precisely that word. Details. The same thing could be said about physiology of neurons, the details of spike timing. It can't be important because it's going to be different from time to time, from neuron to neuron. Yeah, I don't care about the details. I do, but I care about the regularities. And, you know, yeah, that's science. You know, everything is different. Every ensemble of atoms in a gas is different, but thermodynamics works because it goes to a certain place. So, what? Anyway, we'll talk later. This is turning into a.
Just want to comment about this dichotomy between feed-forward and feedback. And so, if you just look at the retina, one of the most salient feedback networks there is the horizontal cell network, which feeds back onto the photoreceptors. And indeed, like, you know, the backbone of the retina is this vertical pathway that goes from photoreceptors to bipolar to ganglion cells. But if you threw out the horizontal cells, that'd be probably throwing out the baby with bathwater for the retina, right? So I think it's just like, I think it could be fundamentally important, but it's hard to kind of, you know, put as good of emphasis on it.
It's throwing out one baby, but it's not throwing out another baby. It's not throwing out the spatial locality of coding. It's throwing out, you know, a gain control mechanism. Bells and whistles. See, yeah.
It doesn't have to be a choice in that you could apply the same analysis.
Yeah, I'm ignoring. I'm doing hemilect to room halves. You pick.
Yeah. So I guess I have two suggestions. One is that if you use the AlexNet, it's similar to our work, that if you use GoogleNet and VGG, would you have the same nice random like the manifold?
I don't know. I was just talking to Michael Bice about this. And musing about very deep networks versus shallow networks. Deep networks with small support, you know, 3 by 3, and shallow networks with larger support, 11 by 11, 5 by 5. And gut feeling, no proof, is that these 50, 100 deep networks with very small support probably get a lot more Baroque in the innards. You know, they probably do. Each layer isn't looking in the same way for the manifold and the layer beneath it. You know, the structures can be mixing layers a lot more. Totally vague, but I don't think it'll work for a deep, narrow network.
It might be interesting to try. And the other thing
this is a hobby.
I really like the kind of narrative and story, but I'm also curious to see, can you give some quantitative metric on how your theory fits the data?
Yeah, one metric that I'm just beginning to play around with is this stress or error signal, how far away the points are from the optimal placement in the manifold. And yeah, you could. In a way, there are a million questions that one can do, but searching for turning this into something that I can assign to people to do.
I have a high-level question. I'm always puzzled in the field of connectomics, like how the community decided to jump from C. elegans, where we have like the complete connectome for 300 neurons, to just trying to map specific, say, cortical columns of mouse or Drosophila, instead of trying to pick like an organism that has, say, 1,000, 10,000 neurons and get a whole connectome. I wanted to have your perspective on that. Why to jump so many orders of magnitude from C. elegans to the mouse?
Our philosophy is, why do I like the cortex? The cortex is a highly, highly conserved computer that is extraordinarily general. And I think the problem of the cortex might be a far simpler problem. That these modules do something. They don't encode this or that or the other thing, and they don't have precisely these characteristics for any given neuron. But in this theory, I don't want to even call it that, they're just an array of neurons that can find manifolds, or find correlations. Let's throw away the word manifold. Find correlations in the inputs, operate on them, transform them a little bit, and send them on to the next level. That's a very simple thing. That the principles might be fairly straightforward. Drosophila is 100 million years of large, many of evolution. Optimizing a small number of neurons. It's probably it's a bag of tricks in much of it. There are parts of the Drosophila that probably are simple in the way I'm calling simple, and might be more amenable to understanding the structure of computation than the highly evolved, highly specialized parts. Very vague answer, but I do sort of deeply believe that the cortex may be far simpler in its generality and more powerful. That's why why we're us.
You should be also doing a cubic millimeter of V2.
Oh, absolutely.
Right,
Yeah,
so
and we're frankly doing the V1, V2 border, but no, exactly. The program is V1, V2. But seriously, Broca's area. Wouldn't it be amazing to find? I won't even say it out loud, but wouldn't it be amazing to find similar principles of finding directions that you can see in the connectome, parameters that encode something that get transformed?
So if there's not more burning questions, I would say we have a discussion section.
I have a burning question.
This is what the complex cells look like, by the way, for anyone who wants to. But no, Dana's been raising his hand forever.
Well, I just want to point out there's a recent, very beautiful experiment with a rat running on a treadmill where the visual input is in concert with the running or disparate. And it turns out that in the thalamus, there are separate buses to take the signal from the things you can predict and the things you can't predict. So this is very in concert. The way I'm thinking about it is that vision is really about prediction, and the only novel things you see are the things you didn't predict. And so and we also know that the feedback connections are 10 times the feed forward. So it's important. It's really important to do what you're doing to get the feed-forward pathway sorted out. That's great. But if you want to see how the thing being used, you have to look at the bigger picture is going to dominate in the outcome.
One thing about putatively powerful techniques like connectomics is you get a class of questions of why not this too? And you make your choices about what to look for. And I would love to do feedback. I've tried to do feedback once or twice. It's just hard. And many people have wrecked their ships of research programs on the shoals of, trying to find, robust phenomena of the neurophysiology of feedback. So I'm conservative.
So if I understand correctly, you're able to go from connectivity to representation crucially because in the visual system, there's like-to-like connectivity. So neurons that have similar representation, similar receptive field, they have similar connectivity. But I wonder if you use your approach to cases where there is not like-to-like connectivity, what insights would you be able to gain? So I can think of several examples. For example, the fly olfactory bulb to mushroom connectivity. So that's random. Or if you might think about your LGN to B1 model, if there's actually not like-to-like connectivity, so LGN neurons that have the same connectivity have uncorrelated receptive fields, what will happen? Or if you have prefrontal cortex where we don't have a good idea of what the representation is, what will happen if you use this?
The first 3 or 4 things, it won't work if the rules are extremely complex. If there is no reliable similarity metric or anti-similarity metric, these tricks are not going to work. But the last thing you said, in prefrontal cortex, you don't know the rules. I guess I have a very strong prior about cortical networks, born out of years of looking at these things, that the excitatory to excitatory connections always, the feed-forward-ish, no, even the lateral. So I don't know about feedback, but the other two, feed-forward and lateral excitatory to excitatory connections, all so far appear to be like-to-like. The network might, including inhibition and different cell types and feedback and all sorts of things, might try to dampen down that like-to-like in order to create sparsity or prediction, etc. But I just, everywhere I've looked and an increasing number of people have looked, there seems to be like-to-like connectivity, and there's Heb, there's B and Poo. That's
my question is more like, so yes, if even in prefrontal cortex you do have like-to-like connectivity, but we don't know the receptive fields of those neurons, what do you get?
Well, that's sort of what I wish I had said more clearly. That's exactly what vision, I understand, and these things are going to be true-ish. They'll be truthy in early vision. Wouldn't it be amazing, and I don't know if this is true, wouldn't it be amazing, is as we go to random cortical networks, even if we don't understand them, we can say, okay, let's play this game. The inputs seem to cluster, seem to look like R2 cross S1. I can do Clay's sort of look and see. I can do nice term I looked up on Wikipedia yesterday, persistent homology. You can do things like that and say, wow, there's a robust thing telling me that the manifold locally looks like this. So I have no idea what these dimensions are, but let's go in and see what the cortex does with it. So that's sort of the general idea, which may or may not work.
Okay, coffee break.